Gene editing is crazy. If you don't know what it is, well then you're in for a treat. Because gene editing is a technology that allows us to manipulate any living organism's gene through a process called CRISPR-Cas9. With this technology, we can literally edit our own genes. We can literally change the way we look by making ourselves taller or shorter, or even change the color of our hair to any color we want. But it isn't just about changing our physical appearance. If we can edit our gene, we can take out any part of our DNA that has a disease or virus, basically curing all genetic diseases. That's right, gene editing could potentially be the cure for cancer. But obviously, it isn't that simple. Because if it was, we wouldn't even be living in this pandemic. The reason why we aren't using this technology today is because gene editing comes with one massive problem. And this problem is off-target effects. Off-target effects is when the Cas9 makes unintended edits to your gene. These changes in your gene can have a massive effect on you because the sequence will then mutate and cause even more diseases or even death. So we know that off-target effects can be really dangerous and what happens if it occurs. But the real question is how? Like, how does it occur? Well, thankfully, we have Ali Haider with us, an expert in gene editing, who is going to talk to us about how this actually happens.
Gene editing is one of the most exciting fields in the world, and everyone knows of its potential, specifically CRISPR-Cas9, or CRISPR with any other Cas nuclease. But one of the biggest struggles with CRISPR is the off-target effects it has. This is because CRISPR is naturally found in bacteria, and it's used as a defense mechanism against viral DNA viruses. And so they use CRISPR to their advantage by taking in specific sequences of that viral DNA from a virus and using it to protect against them. But the problem is, when we use CRISPR in humans or in other organisms, and we're using the mechanism, there are potential for off-target effects because it's found in bacteria. So if there's something similar to that virus that the bacteria finds, it's still going to cut out and destroy that virus, even though it's not exactly the same sequence that it picked up on earlier. This means that off-target effects can occur in insertions, deletions, or mismatches, potentially costing hundreds of millions of lives if not used correctly, and changing the way we even look at gene editing.
After hearing that from Ali, we would probably guess that we still haven't found a way to stop these effects from happening, which is one of the reasons why we aren't using it today. Which means that if we were to somehow find a way to get rid of this problem, we can then be able to use gene editing to not only change the way we look, but to save lives. But what if I told you that there could be a potential solution? As humans, it's almost impossible for us to predict what type of mutations will happen. This is because we can't find any patterns or correlations between large amounts of data. But you know what is good at finding different patterns and correlations between large amounts of data? That's right. The solution to this problem is with artificial intelligence. How, you may ask? Well, we'll be using something called convolutional neural networks, or CNNs. CNNs are used to recognize the spatial information within an image. Now you're probably wondering, wait, an image? But when it comes to gene editing, there aren't really any images. There are only DNA sequences consisting of the letters A, C, T, and G. Well, you're completely right. So in order to use a convolutional neural network, we have to convert these genetic codes into matrices of arrays, making these DNA sequences binary. By creating a matrix of ones and zeros, we will then be basically creating our own image, which means that when the CNN scans these images, it will then recognize different patterns to determine what kind of off-target effect will occur. But before we even start building a model, we still have to get data. This is where the CRISPOR dataset comes in. The CRISPOR dataset is a dataset that includes over 40,000 off-target mutations from the past. After taking the guide RNA sequence and the sequence that was supposed to be edited, the CNN will then find features to see if these images cause either an insertion, deletion, or mismatch. The 3 types of mutations that Ali explained earlier. This is basically how we can create our own AI model to predict these off-target mutations. Gene editing is only at its beginning, and with the help of AI, this technology can help enhance our human existence exponentially, from changing the color of our hair to saving lives.